Hello again, everybody. And we move now to session 2. My name is Rong Tian. I will be the moderator for this session. And I'm a professor at the University of Washington, Seattle. And really excited to kick off this session. And the session was originally planned for cardiac. If you've looked at the agenda earlier, it's called cardiac, but now it changed to 4D metabolism. This is because among the discussions of the panelists, we felt that we should address a broader topic, that is to visualize and target metabolic flux with the spatial and temporal resolution. Basically, this is pretty much the topic left from the previous session, so a really good connection. So I will now ask our first speaker, Zoltan Arany from University of Pennsylvania, to start.
Thanks, Rong. Thanks for the introduction. Thanks for the Vamsi, et cetera, for inviting me. Given the nature of this meeting and the short time, I thought I'd give a slightly different talk. And instead of highlighting our own work, I would just kind of think a little bit at a higher level about metabolism and the cardiac field and sort of what my own personal wish lists are to get this field to advance further. As mentioned, I'm a cardiologist by nature. The heart is kind of a 4D problem. There's no better example. It's continuously beating and it's active and its metabolism never stops, and it's a forward motion that never stops. So we think about that fourth D all the time. And the other point I wanted to make really briefly in the cardiovascular field is that it's not just the heart; it's actually the entire vasculature that matters fundamentally to the cardiovascular field. And the vasculature is a problem because there's really no vascular organ, so it's very difficult to study, and the heterogeneity is tremendous. So I think we really need 3D imaging to improve, to understand the vascular tree. And we need to develop ex vivo tissue models that work. And the same is true for the heart. And that's challenging because there's afterload, there's preload, there's things that need to be mimicked ex vivo. But what I really want to talk about was intermediate metabolism, because when I look at that beating heart, what I think about is all those metabolites rapidly exchanging and going up and down different pathways in order to generate the energy and the motion that you're looking at. And I think there's some really important limitations in our ability to look at this, especially in the intact organism. So this is kind of my very simplistic view of the interest in intermediate metabolism over the last few decades. I think there was a heyday in the mid-20th century and lots of Nobel Prizes and, Krebs and TeSlaa cycle, et cetera. And then it sort of made its way into the textbooks. And I think, 30 years ago, if you ask people what they thought about biochemistry, they thought, well, yeah, I took it in college. And I kind of remember some of it, but not all of it. And obviously there's been a resurgence lately. But, one question that I asked myself a number of years ago is, why did they get relegated to the textbooks? Why did we stop being so interested in intermediate metabolism? And I think the reason is because it got overshadowed by molecular biology. And the reason molecular biology overshadowed metabolism is really because of the toolbox. The toolbox is amazing for us to study molecular biology. And by molecular biology, I mean RNA, DNA, proteins, central dogma type stuff. We have amazing ability to visualize genes with various different northerns and westerns and southerns and PCR and qPCR and immunohistochemistry and some of the images that we've seen. They go from 1D to 3D and now to 4D. But more importantly, we can ask causality questions by manipulating these genes. We can clone them, we can deliver them, adenovirus, AAV, transfection, and we can also go in the organism and surgically alter individual genes with sRNA, with Prelox technology, with now CRISPR over the last few years. And I would argue that in the world of metabolism, we do some of this, but not much. And there's tremendous amount of opportunity in trying to sort of mimic this pathway, this progression. So a few examples, right? So visualizing intermediate metabolism, we're really good in the one dimension, right? We can do mass spectrometry. The technology is getting better and better. The resolution is getting better and better. By resolution, I mean between metabolites. But that's just in the one-dimensional sphere. In 2 dimensions, we're beginning to, as a field, there are technologies such as MALDI that are capable of using the mass spec in 2 dimensions, but it's very limited. The resolution is still very low. It's actually greater than a cell. The holy grail would be, would go beyond the cell and be able to look at subcellular compartments. But that's not there. And this is the sort of thing that can only be done by large collaborative efforts that, for example, Genea could get involved. And then I would argue that in the 3D, and the other thing that Genea could do is expand on take these sorts of things to the 3rd dimension. So for example, we've heard before about the FLY-EM project where electron microscopy is used to Z-stack and create a precise image of the Drosophila brain. I can imagine similar types of things using MALDI, for example, in creating Z-stacks of organs of localized metabolism. This would be hugely useful to the field, to the metabolism field at large, and cannot be done by a single lab, obviously. And then, the 4th dimension, I think, is kind of a relative desert. It's not a complete desert, but it's kind of a relative desert. But we have to tackle it, and if we're able to tackle it, I think we would massively improve research. So there's stuff going on out there. I think we'll probably hear from Gary Yellen about metabolic sensors, and Chris already mentioned metabolic sensors. I think this is one of the waves of the future, where you can introduce a sensor such as an ATP sensor that glows when the ATP to ADP ratio is high. There's NADH sensors that Gary has developed. But that's only a couple. We need to expand that to the large multitude of intermediate metabolism out there. And I think that, again, these are large-scale projects that require innovative collaborations that Genea is poised to perhaps lead. And then the other big challenge with these sensors is to take them to the intact organism. And I think that is also critical. We are particularly interested in metabolism in the intact organism. If we're going to really understand metabolism, we have to take it to the whole body. We've taken one approach to understand metabolism in the whole body, which others have too, but I think is still very, very rudimentary. So we'll do something like you can see this mouse here has a catheter or 2 catheters that are permanently or semi-permanently placed, in one in the artery, one in the vein. And then we can infuse at a steady state level, low levels of carbon-13 labeled metabolites, for example, branched amino acids. Just like Chris, we're very interested in branched amino acids. And then we can achieve steady state, and then we can basically take every single organ in the mouse and look at the contribution to, for example, the TeSlaa cycle, but really any metabolite of carbon-13. And we can create complex maps of how in this case, again, as an example, branched amino acids are incorporated into every single tissue in the body. And these are critical studies, because up to now, these kinds of studies have all been done with either cells or cell extracts, or at best, as Chris had mentioned, for example, a hanging heart. And these are hugely useful, but in the end, they're artificial, and they don't actually reflect what's going on in the body. So I think this is kind of one of the waves of the future for understanding intermediate metabolism at the organism level. But there are severe limitations to this approach still. I think the computation and the modeling is not there. The labeling, the tracing and labeling that you get from these kinds of studies is really complicated and requires modeling to really understand it. There's a lot of opportunity there, I think. The resolution is really low. Obviously, we're looking at, we're mashing up entire organs. If we could take this to a higher resolution, I think that would have a large impact as well. The throughput is pretty slow, and the scale is relatively, so a lot of these reagents are pretty expensive. So there are hurdles here. And we'll hear a little bit more from Tara of these kinds of data. And we've been working a lot with Josh Rabinowitz over the years, and that's been a wonderful collaboration. I'm sure we'll hear from Josh about some of this a little bit more as well. The other approach that we've taken to look at organismal metabolism is to actually put catheters in the organism. So for example, a human, that's a good model. You can put catheters in humans, and you can thread catheters, you can have a catheter in the artery, and you can have a catheter in the coronary sinus, which is draining the heart. And then when you compare what's between those two, you can tell what the heart is secreting or what the heart is taking up. And if you do this with enough numbers, you can get highly statistically significant observations, and then you can do some modeling, and you can do calculations of, for example, what different nutrients contribute to ATP in a heart, and you can compare it to a failing heart. So this is a study we published last year. So this is also very powerful, and this is, I would argue, fourth dimension in the sense that you're comparing what's happening from A to V. So this is a flux study. But obviously, its resolution is, again, low, and the throughput is super low. And the other problem is you can't, this is sort of the opposite scalar problem. You can't do this in small animals. You can't put catheters in the coronary sinus of the mouse or arteries and veins of the mouse. So these kinds of studies, if you really want to take them, if you want to go at them full on, I think you have to go to bigger organisms than the mouse. Chalsoon Jang, who used to be in my lab and then joined Josh's lab, while he was in Josh's lab, started doing this kind of thing, looking at a pig and instrumenting the pig in multiple different vessels at the same time, and then doing what I just showed you for the branched amino acids what I just showed you for the heart, and that works, but this is just the tip of the iceberg. These are hugely expensive studies, and again, studies that need to be done, if done well, are going to have to be done by large collaborative groups. So I think there's a tremendous amount of opportunity here as well. And then the last thing I wanted to talk about is the bottom part here, the sort of, the ability we have to surgically manipulate genes and go in and knock down one gene and only one gene. We don't have that in metabolism. We cannot create a metabolic knockout, or hardly can. Could we go in and figure out ways to delete individual metabolites or lower individual metabolites and study the impact on the rest of the organism or the rest of the metabolic network? I think would hugely advance the field. There are a couple examples out there, and actually, Vamsi has in some ways led this field. His lab has created, for example, usurped bacterial enzymes to take NADH back to NAD without having a large impact on the system, and that allows you, surgically to analyze the impact of just doing that, or for example, just converting lactate back to pyruvate and changing that ratio. But again, this is just two examples in what needs to be, thousands and thousands of examples. So there's tool development here that I think really needs to be done, and would be very, very useful. And again, same question as before: how do we take this to an organism? Obviously, we try to create transgenic mice that express this in particular tissues, for example. These are the sorts of things that I think are worth thinking about. So that's kind of it. I think I'm a minute or over time or so, but I think we can sort of leverage the amazing impact that these precision tools had for the molecular biology revolution, and see if we can design sort of similar precision tools and imaging technologies to do the same thing in intermediate metabolism. And I think that would really allow us to accelerate what we're already doing. I'll stop there.
Thank you so much, Zoltan. And if there's any questions from the audience, please type into this Q&A box. And the panelists, if you want to ask a question, use that as well, or you can just shout. Maybe I can get started. And I think so. You framed those questions are really nice, and especially talking about what can be done in animals and the cell culture models versus what can be done in humans. Do you see how those results can be integrated? We often hear that humans are not animals, and patients don't model mice. How do we address that?
I think ultimately it's depends a little bit on what your question is. as a physician, I tend to think that ultimately I want to know about people. But I think there's also value in just asking questions about fundamental biology. Not everything has to relate to clinical medicine. So I'm perfectly comfortable asking questions about how does a worm work without necessarily having to have it impact, human physiology. But having said that, I think the integration of those integration of what you learn in the one organism with what you learn in the other organism is hugely insightful. How do we do it? Again, involves large collaborative groups and a lot of deep thinking. I don't know if that answers the question, but
yeah, that just means more work to be done.
Yeah, absolutely. There's no doubt about that.
Which is wonderful. So maybe we will save this for more discussion during the panel, and then we can move to the next speaker, which is Jared Rutter from University of Utah. Jared, it's yours.
All right, thanks, Rong. And I'm also glad to be here. And a bit like Zolt, my 10 minutes or so will be spent at a pretty high level. I've actually had some time to think and talk with my lab about where I think the field of metabolism is and where it has been and where I think it needs to go. And so my comments are going to be pretty high level and conceptual. Like Zolt, I've thought a lot about the molecular biology revolution that happened, and many of us think of it as having kind of destroyed the field of metabolism back in the 70s and 80s. But one thing I think it did do is it caused the study of metabolism to change from a study focused on metabolites to studies focused on genes and gene expression and protein signaling, et cetera. And I'm sure those of you that are old enough will resonate with this. When you go to a meeting and there would be a metabolism session, maybe in 2000 or so, all the talks would be about transcription or signaling. And they would be about, the genes and proteins that do metabolism would be the targets of those transcriptional or signaling events, but it would all be about transcription and signaling. And it's been, I think, gratifying to see that in the last several years, catalyzed by many people that are in this workshop, the study of metabolism has changed in many ways, or evolved forward to include some of the concepts that were maybe important 50 years ago. So I guess the message that I have is that as we move forward studying metabolism, let's not forget that metabolism, in my view at least, really means the metabolites. They're the key elements of metabolism. And so we're going to talk in this workshop, I think, about all kinds of very powerful technologies that can and should be developed to help us understand metabolism. And that is absolutely fantastic and awesome, and we need to go there. I think there are also sort of fundamental and foundational issues that we need to be thinking about as well. And in a way, maybe not repeat what happened in the past, where we maybe lost some perspective on what were the key questions. As we move forward, let's maintain our focus on, like I said, sort of foundational issues. So I've been thinking about what do we need to know about metabolites in order to understand them in this four-dimensional cellular physiology context. And I'm going to tell you what I think those questions are, and then maybe just really briefly dive into some of the things that some detail about each one of those. And I think I will talk about them in varying degrees, because some of them I think are more obvious than others. So clearly, if we're going to understand metabolites in the context of cellular physiology, we need to know what chemical reactions those metabolites are engaged in, to what are they being converted, and what are the rates of those conversions. I think we need to know where is the metabolite. And as you'll see, I'll talk about this a little bit more. I think there are all kinds of different spatial resolutions that we're talking about here, all of which I think are important for understanding cellular and eventually organismal physiology. And finally, what does the metabolite do? And that may seem like a trivial question at first, but I'll talk a little bit about what I mean by that. So to focus on this first one, I think it's pretty obvious, and I think most of the speakers that have talked about this, that I think we need continued development of metabolic tracing and flux analysis. In my view, the ability to measure metabolism quantitatively over time is exceedingly important, and we have dramatically increased our ability to do that. And to some extent, this technology has been democratized, where it's not just 2 labs in the world that can do this now, but it's being done widely, and in many cases being done well. And I think that needs to continue to happen, and the developments in that area need to continue to emerge, both with resolution and depth, like Zolt talked about. And also, as Zolt talked about, I think it's becoming more and more important that these experiments are done in the context of systems that are more physiological, and either more physiological in vitro systems or even in vivo. And there are several people in this workshop who have done metabolic tracing in humans now, and allowed us to have a reasonably good surrogate of metabolic fluxes in vivo in a tumor, and through collaborations that we have in the heart, and other people have done this as well. And I think this is a really exciting development. And I think these are all areas that I think need to be continued to be advanced. And I agree with Zolt that the development of sophisticated collaborative teams is important to move this forward, because it is expensive and complicated, and the modeling involved is demanding. Second question, where is the metabolite? And this is asked in sort of a qualitative way, but I think this is really a quantitative question. Basically, the question is, what is the abundance of a given metabolite at each place in the context of an organism over time? And I realize how absurdly complicated that question is. And we're a long way away from being able to answer that question. But I think, in my view at least, that's where we need to go. That is the frontier that we need to cross to get to a true understanding of 4D cellular physiology from the context of metabolic physiology. And we talked about, Zolt talked about this in the context of organs, and that's certainly important. What are the fluxes across organs? But I think we also need to understand this at the level of single cell resolution, and even at the resolution of individual compartments within cells, individual organelles, for example. I think we all recognize the fact that NADH in the cytosol and NADH in the mitochondria means something that's very, very different. And we need to understand the abundance of those to truly understand how the cell's behaving. And we understand there are many metabolic pathways that are shared across across cellular compartments. 2 examples are shown here. And I think we understand the importance of those, that shared metabolism in driving cellular physiology, and understanding where metabolites are and what they're doing, I think is really important. But maybe something that I think is less well appreciated, including in our field, is the degree to which this might be happening in an intercellular way, locally. And in my view, kind of the foundational hypothesis related to this is the lactate shuttle hypothesis. This is a figure published by Pierre Magistretti, who I think has been a major proponent of the lactate shuttle hypothesis in the brain, whereby wherein glucose is taken up by astrocytes, oxidized through or converted through glycolysis and lactate dehydrogenase to lactate, which is then excreted and then taken up by neurons, which then convert lactate back to pyruvate and oxidized in the mitochondria. This is one example, and this is presumably very important for the physiology of this little microenvironment in the brain. I think it's very likely that this is happening all over the body. In virtually every microenvironmental niche, there is this kind of metabolic communication, not always related to lactate or glucose or any carbohydrates at all, but is probably happening all over. And we need to be able to understand, have the resolution to be able to understand which cells are doing which forms of metabolism. And I think spatially resolved metabolomics, which we have and will talk about extensively in these 2 days, I think is one tool there. Zoltan mentioned biosensors. I totally agree that a generalized suite of tools that would allow us to measure many metabolites or eventually most metabolites in their physiological concentration ranges would be enormously valuable for helping us understand where metabolites are quantitatively. And I think that one of the foundational issues in dealing with this is that metabolite transporters are the proteins, are the elements that move metabolites from one place to another, either within cells or between cells. And I believe that one of our major deficiencies in understanding metabolism physiologically is our profound lack of understanding about metabolite transporters. It's my view that this is probably the most poorly understood, poorly annotated family of proteins that act in metabolism. And that comes from the fact that the assays that we have are simply awful. They're bad. The small number of people that study metabolite transporters in the world typically are using assays that are very, very similar to what was used in the 1970s for studying transport reactions. And I think we need major improvements there to understand the proteins that move metabolites. And finally, the question, what does the metabolite do? And I want to focus just really briefly beyond just the role of metabolites as intermediates. And we all know that metabolites have signaling roles, and there are now keystone meetings focused on the signaling roles of metabolites. So there's really been a growth in the understanding of that field, but it goes beyond the roles of metabolites that we conventionally view as signaling, like acetyl-CoA, which is a substrate for acetylation and so forth. If you ask somebody in the 1980s what signaling metabolites look like, they'd probably describe something like this, where a bunch of metabolites signal into control the enzymes that do glycolysis and gluconeogenesis. I think this is what sort of the ancestral signaling role of metabolites looks like. And this is a very small subset of just a signaling that goes in just to glycolysis and gluconeogenesis. And I hesitate to show this slide because it emphasizes what I think is a false impression, that metabolites only signal to metabolic enzymes. I believe, and we know many examples, and I think, but we only know the tip of the iceberg, that metabolites signal to everywhere in the cell. All cellular processes, in my opinion, are subject to metabolite level regulation, most likely, including metabolism, like I talked about, also transcription and signaling, which we know many examples of this happening, and I think there are many more, and also aspects of cell biology that we don't typically think of as being metabolically regulated. I think many of these, or all of these, are going to be responsive to metabolism and metabolic signaling in some way or another. So I think one of our, again, foundational issues that we need to understand biochemically before we can put it into the context of organismal or even cellular physiology are the metabolite and macromolecule interactions. What are they, and what are the effects that those have on those macromolecules, and then eventually into the biochemical pathways and cellular physiology that we all care a lot about. And so I'll just end there and say, I think we are in the midst, and I think are on the precipice of revolutions related to imaging and and metabolite measurement. But I think we need to keep the foundation strong of understanding the biochemistry that will help us understand that physiology. So with that, I'll just thank you and take any questions that you have.
Terrific. Thank you so much, Chair. That's really thoughtful. If anyone have questions, please enter it into the Q&A box. I see Ashley raise her hand. Go ahead, Ashley.
Hi, Jared. I'm always grateful to hear your thoughts on all things metabolism. I wonder, with regards to the idea of metabolites interacting with macromolecules, to what degree you think that has an effect on, say, sequestration of metabolites to change their concentrations in given compartments and whatnot. I'd just like to hear your thoughts on that.
Yeah, that's a great question. You know, I talked about metabolite macromolecule interactions as being a way that metabolites could regulate macromolecules, but maybe it's the other way around as well. Maybe macromolecules can sequester metabolites, and there are some limited examples of that. I think that's absolutely a possibility, especially when you think about structures like liquid-like droplets, for example, or stress granules, where there are polymers that you could imagine titrating metabolites significantly. So I think that's a really intriguing idea. And you know, when we think about compartmentalized metabolism, I think we obviously have to think about those structures as well, in addition to the membrane-bound compartments that we tend to focus on when we think about metabolite compartmentation.
I want to follow up on this signaling role of metabolites. I think, you know, the signaling role of proteins made progress so well because we discovered how to target kinases and phosphatase and all the protein modifications, and we have ways to study protein-protein interactions. So how do we do that for metabolites? How do we target metabolites? I think it's similar to what Zoltan also mentioned.
Yeah, I think I'll give two answers to that question. And one is an unsatisfying one. As Zolt said, it's really hard. You can't make a knockout. You can't knock out the gene for glucose or for, you know, alanine. There's no such thing. And even if you could, the cell would adapt and fix it, or at least adapt to it. So I think that kind of, you know, pseudogenetic kind of strategy, I don't think we're there yet. Obviously, that is, again, sort of it has to be one of our goals, is the ability to manipulate metabolism specifically and acutely. But I think one other answer to your question, Rong, is that if we view signaling as being driven by physical interactions, one way that we can go is by understanding the interactions that metabolites make with proteins. And my lab is doing work in this area. There are many others that are as well. And I think, you know, we are really, really good at understanding the interactions between many of the kinds of cellular molecules, but that does not include metabolites. We are relatively poor in discovering or characterizing or understanding the interactions between metabolites and everything else. And I think we just need to develop the tools and implement the tools to do better there.
Great point. So we have a question from the audience. Amy Elliott asked, do you have any thoughts on using simpler model organisms for studying the evolution of complexity with respect to metabolite regulation and communication between cells?
Yeah, that's a really good question. Absolutely. I mean, my lab uses yeast as a model system, so I'm very comfortable with simpler model organisms and love the power that one can obtain from using simpler model organisms. When we're talking about multicellular communication, that obviously puts some constraints on that. But I think there are great things that can be learned. I mean, Chris talked about this with organoids, and obviously the islet is a unique situation, But my guess is that there is metabolite-based communication going on in the islet that is controlling hormone secretion and signaling and so forth. And the ex vivo islet might be a system that could be used to study that. I don't know. I don't know much about it, but I could imagine that it might. And certainly simpler model organisms probably recapitulate many of the microenvironmental niches where there might be intercellular metabolite signaling between neurons and astrocytes, or between, we were supposed to be talking about the heart. I think this is obviously going on in the heart as well.
Yeah, and maybe engineered tissues, even bringing indicators and sensors in a multicellular structure.
Absolutely.
Jennifer has a question. Jennifer, go ahead.
Yeah, sure. That was really great. Love hearing you talk about all of this stuff. I'm curious as to, we talk, when I hear you talk, the perspective that I'm getting is that the cells are sort of in a particular state, and we're trying to figure out how these metabolites are distributing and behaving. But cells crawl, they go into polarized arrays, and we know from prior work that there's hugely different types of metabolic signatures that correlate with these different cellular behaviors. And so I'm curious as to how you think about that from the perspective of metabolism. Who's driving who?
Yeah.
Is the cell's particular desire to crawl or to become part of a polarized array dictating the metabolism it chooses? Or, does the metabolism drive you to a particular state? Just how is this is part of a sort of a larger perspective on control of the systems, but basically metabolism drives physical processes within cells. Actin, polymerization needs ATP.
Right.
How is all this connected to what the cell is actually doing?
Yeah, you kind of scratched probably my deepest passion, my deepest scientific passion is we've always thought about metabolism being subservient to the cell, to the rest of the cell, that it just does what it's told and you give ATP and it'll do it or whatever.
Exactly.
I think the evidence is growing dramatically, and it's growing by the week, that metabolism is doing more than that and is actually controlling whether a cell crawls or not. It's not just providing the ATP that a cell needs to crawl. It's controlling the decision to crawl. And I think, again, in my view, that one way of viewing that is the signaling effects of metabolites on the decision-making apparatus of the cell. And it's not one apparatus, it's many apparati. And to the extent we understand them and how signaling feeds into that, both metabolite-based and otherwise, I think that's when we really start to understand metabolic physiology at the level of the cell, in my view.
Yeah.
Could I add something to that really quickly? Maybe simultaneously eat into the 20 minutes from later.
Yeah.
Because I completely agree with that. And I think, any geneticist will tell you that the way you prove causality is by doing epistatic experiments or something equivalent. If you really want to know what's causing what, you have to intervene in the system and see what the consequences are of your intervention. And I think that was one of the points I was trying to make, is that our tools for doing that with metabolism are not that good. I mean, sure, we can throw a metabolite at a cell monolayer. Maybe this metabolite will get in, maybe not. Maybe we have to modify the metabolite to get in. We don't really have any good tools for removing a metabolite from a cell. So I think, it would behoove us to think that way, to think about how can we precisely intervene in the system in order to study the outcomes. And that'll tell you what it's regulating.
Right.
I might also add one last point to that, if that's okay. And that is, speaking of what drives what, it could actually be thermodynamics. If we think about the biophysics and laws of physics, maybe the thing that really governs how the cell is able to harness mitochondria and how other processes may be limited by the mechanics as well as the thermodynamics that's involved.
Okay, so we're running over time. I hope Jenny is okay with
All right.
this. And we pre-show the discussion part right now, but let's get back to the presentation. So the next talk will be Vijay Rajagopal from University of Melbourne. Vijay, please.
You're muted, Vijay.
Thanks, Ron. Are people able to see me? See my slides
Yep.
here okay? Or
Yes.
okay, fantastic. It's just telling me to unmute myself. Okay, great. So thank you, everyone. And thanks to Geneva Research Campus for giving me this opportunity to speak. And thank you also to Jared and Zolt for great stimulating opening discussions, actually. It was really fun to hear you guys speak. So today, I just want to very briefly discuss the work that we're doing in my research lab at the University of Melbourne. And I always open up with a starting slide here, which is not from my lab. It's the work that actually inspired me to get into this space. What you see here on the left are confocal images of cardiac tissue architecture. A being a control wrapped tissue architecture, B is diabetic. And then on the right are these transmission electron microscope images of the ultrastructure of the cardiomyocytes within these tissues. So you can see the myofibril striations, and the darker spots are basically mitochondria. And you can see in diabetes, this organization is affected. And what these guys did was they applied a copper chelator as a potential treatment, and they found that the architecture of the tissue changes, and so does the architecture of the cell itself. So while we talk a lot about signaling and the role of biochemical reactions in regulating cell function, structure is inherently tied to all of this. And so my lab's really interested in understanding how structural organization is also feeding into cellular physiology. And today I'm just going to show you how we use computer modeling to investigate this aspect of cellular physiology. So what I did in my lab is we also collected some electron microscope images from these are streptozotocin-induced diabetic rats. And what you're seeing is cross-sectional images. So if you think of a cardiac cell as a cylinder, you're looking at the circular face of that cylinder. And what you'll see are the dark spots are basically individual mitochondria that we've identified, manually annotated, and it was a green boundary. And then the rest are basically myofibrils and sarcoplasmic reticulum and things. So the left is a cardiomyocyte cross-section in a control animal, and the right is a diabetic one. And in work that we did to quantify the changes to mitochondrial organization, we basically quantified that in diabetes, we found that we form lots more smaller mitochondria that were all clumped together, still maintaining that overall columnar architecture that we're so familiar with in cardiac cells. So this is kind of representing sort of a fission state, if you will, but these are electron microscope images, so we don't fully know exactly what's gone on to get to the state here. The other question we have here is, well, what is the influence of this change in architecture to the function of the cell? And that's a very engineering question in a way, because engineers, we like to look at structures, and then we design structures, and then we put these things together and simulate some function on it. Like, if you're thinking about building a building, is it strong enough? Well, let's see if I put a few pillars here and there, will it work? That kind of thing. So if you think about it that way, you've got these mitochondria producing energy, and they need to that energy needs to be distributed through the cell. What is the impact of the change distribution on these on the energy distribution? So there has been a sort of a change in our perception of what mitochondria do inside cells, at least up to say 20 years ago, maybe 15 years ago. We all traditionally thought that mitochondria are basically these lozenges that are sitting inside the cell, that they're just producing energy in isolation. But more and more, at least it's certainly in the last 5 years that I've been working in this area, but that's definitely been, at least for the last decade, we're starting to see how mitochondria are more dynamic. They form these 3D organizational networks. There may even be electrical coupling between mitochondria that drives energy metabolism. And so what we're interested in doing is creating a computer model in which you can see how these different characterizations of what mitochondria do influence each other and govern energy dynamics. So in this work, what I'm going to show you first is a crosstalk that we've looked at between how the OXPHOS system works and how the mitochondrial organization works, how the two of those may be regulating cardiac function. And then I'll briefly mention mitochondrial dynamics. So in order to study this question of how architecture and oxidative phosphorylation are interacting in the myocyte, what we do is computer modeling. And there are quite well-established parameters, and we very well know the reaction pathways involved in oxidative phosphorylation. So you can convert these reactions into a set of ordinary differential equations, what we call mass action kinetics models. And when you bring those together into a system of ODEs, you can then have this model of mitochondrial oxidative phosphorylation that represents the electron transport chain reaction pathway. We can optimize parameters in that model for either control conditions or diabetes or any other disease condition we may like to look at, as long as we have some functional data from seahorse measurements and things related to that. And then what we do is look at also coupling this model of oxidative phosphorylation with a spatial model of the diffusion of metabolites. So this is that where question that Jared was pointing to before. Here what we do is we can take our spatial image, this electron microscope image of the cell, and then we can simulate reaction diffusion processes on it using partial differential equation simulation techniques. So in this model, we've included the ATP, ADP, AMP, as well as creatine kinase, which is considered as a shuttling mechanism for rapid transport of metabolites. And then we take the images that we have. These are electron microscope images, 2D as well as 3D serial block phase images. And then we can create what we call a finite element mesh. These are computer modeling techniques that people have used in Boeing and many other fields in computational bioengineering. And then start to visualize the coupling of oxidative phosphorylation function with transport of metabolites within that mesh. So here you're seeing, this is a cross-section again of a cardiac myocyte, and then these are images of the mesh in high resolution that you're going to see the results of next. So here's what, so this is that original image of the cross-section. I'm able to simulate things like ADP to ATP ratio, inorganic phosphate distribution within the cell. You can see it's fairly uniform through that cross-section. And I can compare my model predictions against experimental data. Here you see the blue dots representing my model predictions of PCR to ATP ratio relative to oxygen consumption. And the red dots are experimental data. This is on a whole cell assay. The bottom one is a more individual cell spatial assay, where the red lines represent oxygen concentration through the cardiomyocyte, and the blue lines are our model predicted ones. And you can see that just by coupling together the biophysical models of OXPHOS and transport of metabolites, we can start to simulate the metabolic physiology in silico. And what we can do with that then, with this model, is we can start to perturb it to see what would happen inside the cell, something that we're not able to do right now at high resolution. We can start to do with these models. And indeed, as Jared was saying, and Jared and Salt were saying, we need to get further into making high resolution measurements, which will then further inform these models and give us a better picture of what's happening. But at this stage, you can see by just reducing the oxygen consumption from a high oxygen level to a low level, I can see how the membrane potential here, represented on that cross-section, mitochondrial membrane potential, starts to have a spatiotemporal heterogeneity. You can see this inner region through in the thickness of the cell has a lower mitochondrial membrane potential than on the cell boundary because of that reduced oxygen supply. And you can also look at complex 4 activity, complex 5 activity. You can also couple this oxidative phosphorylation model and metabolite model with cross-bridge kinetics and look at how mitochondrial function and the organization of the mitochondria affects the contractility of the cell. And again, with this low oxygen supply, you see this anoxic core where there is a greater, a longer twitch duration in the cardiomyocyte. So you can start to see there's potential shear forces being developed, just minor amounts as predicted here, but nonetheless could be important for what the cell does next in the long term. So the final slide, and I'll just very quickly do this. We also looked at how this organization that is affected in diabetes impacts on cross-bridge kinetics and force production. And so the left-hand side are cross-sections of control myocyte. The right-hand side is a diabetic myocyte cross-section. And you can see heterogeneity in the metabolite distribution within these, within the diabetic one. And when you look at the distribution of forces produced across that cross-section, the blue one represents a control myocytes cross-bridge kinetics, just force production. And you can see it has a high peak, saying that it's fairly homogeneous. But in the disease state, it's much more where the structure is disorganized of the mitochondria. You can see a broader distribution of force production within that cell, clearly showing that structure has a significant impact on what the cell is doing subcellularly. So with that, I'm going to finish with just a couple of points. That is to say, what I've shown you today is how we can use computer modeling to interrogate how structure and function are related in the subcellular space. What I hope you can also see is that by using computer models, we can look at more than just a couple of points. You can start to tie biophysics with measurement to make mechanistic predictions and understanding of what's happening in the cell. And the last slide is that the dream experiment or model that one may want to do, and certainly I'd like to see done, is more precision measurement of subcellular spatial metabolism, higher precision localized perturbations of metabolism, and that question that Jennifer raised before, how is the cell harnessing mitochondria or vice versa to drive cellular physiology? That's the question that I'd like to answer with the kinds of models that we can build. Thank you very much.
Thank you so much, Vijay. We're a bit behind, so if there's any burning questions from the audience, please type it in. Otherwise, I will save all the panelists' discussion to the discussion session. Okay, I see no questions from the audience. Let's move to the last talk by Tara from Princeton University. Tara, please go ahead.
Hi, thanks. Thanks to Rong, and thanks for inviting me to share some of my postdoc work in Josh Rabinowitz's lab. And I think I'm excited to share some of this data because it shows some of the trouble that we have with trying to measure metabolic fluxes in vivo. So everyone here knows that glucose is a very important circulating nutrient. It's the most abundant circulating carbohydrate, and it's a very important energy source, especially for the brain. What's becoming more appreciated is that lactate is also a very important circulating nutrient, And our lab and others have shown that lactate is both produced and consumed at a very high rate on the whole body level. And I became very interested in this because since lactate is the product of the glucose catabolic pathway glycolysis, this has a lot of big implications for glucose metabolism. And we know glucose needs to be maintained at a very tight range of concentrations in blood. So I became interested in understanding where the circulating lactate is coming from as a tool to deepen our understanding of glucose metabolism. And so in this presentation, I'll talk about two different questions related to this. How much of the circulating lactate is coming from circulating glucose versus stored glycogen? And then which tissues or cell types are doing this glycolytic flux? So to start to look at this, I infused carbon-13 labeled glucose into jugular vein catheterized mice, just as Zolt described previously. And what we can see in these mice is the appearance of our labeled metabolite, which is glucose, over time. And as we infuse this labeled glucose, we can quickly see labeled lactate, the product of glycolysis, appearing in circulation. And by the end of this infusion, what we can see is over 50% of circulating lactate being labeled by glucose. So we can conclude from this experiment that more than 50% of lactate comes from circulating glucose. But how much comes from glycogen? So to do this, we need an experiment where we can use, instead of labeled glucose, we can have labeled glycogen. This is a little bit tricky, but we did this by doing a pulse chase infusion of carbon-13 labeled glucose. So we infused glucose in a pulse for 16 hours, during which time this carbon-13 labeled glucose is incorporated into tissue glycogen. And then we do an 8-hour chase. During this time, because we're not infusing labeled glucose, the labeling in circulation should decrease, but we should maintain the labeling in tissue glycogen because of its slower turnover. And so now we have a system where we have labeled glycogen and we can ask how much lactate comes from that glycogen. And so what we see when we normalize our labeling to glycogen in the tissues is that we have a large amount of labeling of both circulating glucose and circulating lactate. And this makes sense because we know that a lot of glucose comes from glycogen in the liver. So the question becomes, does this labeled lactate come from the labeled glucose or the labeled glycogen? But we have prior knowledge that in our short glucose infusions, when we infuse glucose and glycogen is not labeled, that we have about 55% of lactate labeled relative to glucose. So we can use the information from these two infusions to actually calculate the fraction of lactate that comes from each of these sources. And so what we find is that around or a little bit more than 50% of circulating lactate comes from glucose, and a smaller but substantial fraction also comes from glycogen in the tissue. And because we know the lactate turnover rate, we can convert this measurement into a flux. So what we can see is that the flux of glucose to lactate conversion is very large on the whole body level. And so this brings me back to my point, my second question: where is this flux happening? Because we know that this is a very large flux. The cell or tissue that is doing this conversion of glucose to lactate would presumably be very important for maintaining glucose homeostasis. So to look at the tissue or cell type, we had a very simple idea. When we infuse labeled glucose, we can see labeled lactate in circulation. So the tissue or cell type that is producing this labeled lactate would presumably also have labeled glycolytic intermediates. And if a tissue or cell type is not using circulating glucose, but is instead using glycogen, well, in our short glucose infusions, glycogen is unlabeled, and therefore the glycolytic intermediates of that tissue would be unlabeled. So we infused glucose, collected tissues, and looked at labeling of glycolytic intermediates. And here I'm showing 2 intermediates, fructose bisphosphate and 3-phosphoglycerate. And what we saw when we looked at tissue was not quite as black and white as what I described earlier. We see low to intermediate labeling and glycolytic intermediates in these tissues. And so we can't really confirm that these have this high lactate production flux that we expected. However, when we expand our search to a lot of tissues, so next I'm showing the average labeling of glycolytic in several tissues, we can find some tissues that label very highly from circulating glucose, or at least as high as circulating lactate. And so 3 tissues here on the left, we can see whole blood, which is mostly red blood cells. So this makes sense because red blood cells don't have mitochondria and should be using a lot of glycolysis. But we also see 2 muscle groups, so diaphragm and soleus, and I'll talk about these a little bit later. But another question we could ask is, where do all of these other glycolytic intermediates come from if they're not coming from circulating glucose? And an obvious place that I talked about previously would be glycogen. So we did pulse chase infusions to measure the contribution from glycogen to these glycolytic intermediates and quantified their direct contributions. And so what you can see is in several tissues, glycogen is a major source of glycolytic intermediates. And in a lot of these tissues are very large and have large glycolytic intermediate pools. So when we add this up on the whole body level, we can see that most glycolytic intermediates in the body come from glycogen. So this seemingly is kind of a paradox where when we look at glycolytic intermediates in tissues, most come from glycogen. But when we look at circulating lactate, which is downstream of glycolysis, most circulating lactate comes from glucose. So what's happening? Well, what we think is happening is that glycolysis is compartmentalized. One place we know that it's compartmentalized is in these muscles that I talked about earlier. However, based on AV data that I don't have time to discuss today, we don't think this is responsible for all of the glycolytic flux in the body. So what we think is happening is that glycolysis is also compartmentalized into specific cell types within heterogeneous tissues. And as many people have already discussed, when we do standard LC-MS measurements, we take chunks of tissue, grind it up, extract the metabolites and run it on the mass spec. And we lose the spatial and the cell type specific information about metabolism. So we need a tool to start to dissect this heterogeneity of metabolism in tissues. And when we think about glycolysis, we can imagine that some cells within a tissue may have very large glycolytic flux and use a lot of circulating glucose. However, if many other cells in the tissue are using glycogen to do glycolysis, the labeling of or the lack of labeling of these glycolytic metabolites will dilute out our signal. And so we want a way to try and identify these glycolytic cell types. What we've started to use, and many people have discussed this previously, is imaging mass spectrometry. So this is MALDI, and we're using it in collaboration with a fellow at Princeton, Sean Davidson, and this is another postdoc in the lab, Lin Wang, who's been helping me. And so what we can do is we can take tissue slices and put a matrix on top that allows us to ionize metabolites within the tissue. And we shine a laser to ionize those metabolites. They go into the mass spec. And because we can do this several times, we can create an image of metabolite abundance across the tissue. So I wanted to use this to identify glycolytic cell types within tissues. And again, what I'm looking for is just cell types that have labeling from glucose in their glycolytic intermediates. And I decided to start doing this in muscle as kind of a test case, because we know that different types of muscle label very differently from glucose, where the soleus labels a lot from glucose and the gastrocnemius muscle, which happens to be right next to it, labels very lowly. And I can take a section and stain. This is immunofluorescence. And so we can see that the soleus is mostly composed of type 1 and type 2A fibers, these slow and intermediate fibers, while the gastrocnemius muscle is mostly composed of type 2B fibers, which are more fast fibers. And so likely the differences in metabolism that we see are differences in the muscle fiber composition of these tissues. But what we can see also is that the gastrocnemius muscle has this inner region, which we refer to as the red gastrocnemius, because it has more of these intermediate and slow fibers. So we hypothesize that if we use MALDI, we could see differences in metabolism across this tissue. And so here's some imaging mass spec data. And what I'm showing here is fructose labeled, the fraction of fructose bisphosphate that is labeled from glucose. And so what we can see here is in the soleus, most of the fructose bisphosphate becomes labeled. And this matches very well with our LCMS data. And we can also see that gastrocnemius muscle has much less labeling. But what we can also start to see with MALDI that we can't see with LCMS is that there is more labeling in the redder part of the gastrocnemius muscle and less labeling in the white. And we can draw regions of interest here and quantify this and see these differences in labeling. So we think that imaging mass spec is one tool that we can use to start to dissect the heterogeneity of metabolism in tissues, although a lot of work needs to still be done to improve the resolution, as many other people have discussed previously. So in conclusion, glycolytic flux is compartmentalized into specialized cell types, and improved tools such as imaging mass spec are needed to visualize or dissect the metabolic heterogeneity of tissues. So I'd like to thank my lab and thank you for your attention.
Wonderful. Thank you, Tara. So a couple minutes for questions from the audience. And then we'll, I see questions from the panelists. And so I don't see any questions from the audience. So I go with the panelists, then Chris first, then Wamsi.
Yeah, that was a beautiful presentation. Thank you very much. Many moons ago, with Dennis McGarry, I studied glycogen metabolism in the liver, and we published a series of papers about an indirect gluconeogenic pathway being particularly important for glycogen synthesis, particularly in the early stages of a fasted to fed transition. Okay, in other words, there's quite a lot of gluconeogenic substrate flow, including lactate, to form glycogen, especially in the early stages of the fast to fed. So I just wonder, is there a potential for some overestimate of the contribution of glycogen to lactate? Because if you're giving labeled glucose and it was converted to lactate, could that lactate have been used to synthesize the glycogen, which is then used to synthesize, to generate the labeled lactate again? Do I make myself understandable?
Yeah. So actually, I didn't show it here, but I have also done lactate infusions. And so we can measure the contribution of lactate to glycogen and to glycolytic intermediates and use our same like mathematical approach to dissect out these different contributions. And so after correcting for that, it wasn't a huge difference in the contribution, but we definitely could be overestimating the contribution a little bit of glycogen to glycolytic intermediates in several ways. These are hard measurements to make. And so, yeah, one way could be that the glycogen molecule is not homogeneous. And in our measurements, we can't tell the difference between like the outer shell or the inner shell of glycogen.
Okay, thanks.
Thank you, Wamsi. Quick questions.
Great, Tara, a great talk. So what fundamentally limits the spatial resolution? Is it the low abundance of the metabolites, or is it the nature of the excitation?
I think it's a lot of things. So definitely the amount of the metabolite is more limiting when you go to a higher resolution, right? So you just have less, the smaller the laser spot gets. Then like we're limited by the actual size of the laser. So the smallest laser spot that we have on our instrument now is about 20 microns. And then one more thing that you might not think of is the matrix. So the chemical that we deposit on top of the tissue actually forms crystals. And the size of those crystals determine how much metabolite will be ionized with each laser spot. So that's another area that could be improved.
Thank you. And there was a question in the Q&A box from the audience. I missed. I think it's from for previous speakers, but I bring it up since the audience has no chance to ask question when we move to panel discussion. So this question is asking about basic chemistry in subcellular compartments, such as pH regulation. Are there any advances to have a better control of that? So anyone has any thoughts on this?
I mean, this gets back to the question of precision disruption of the system. I think it's a really excellent question. You know, one could imagine, let's take the pH example. One could imagine a genetically engineered protein that somehow alters local pH, and then delivering that to, you know, the mitochondrial surface or to the plasma membrane, and then seeing what the impact is on cellular physiology. I think these sorts of approaches are ripe, and we have the capacity to do them, I think, but they'll definitely take some concerted efforts to achieve it. But I think they will answer, you know, they're fantastic questions.
Great. So there comes up a question in a Q&A box from Brian Clancy. He said there are increasing number of probes that can especially resolve the concentration of metabolites with cellular or subcellular precision, but there are few, usually very complex ways to visualize flux of a given metabolites at similar scale. So what would it take to make flux probes rather than concentration probes? I think this is a great question.
Yeah, this is a really hard question. And I think, I don't know that we will ever be able to have biochemical probes that measure flux, you know, like the equivalent of the concentration probes that Brian mentioned. If somebody has an idea for that, I'd love to hear it. That would be super cool. I would imagine that flux probes are going to have to be chemical. Gary just raised his hand and he's the guy for probes, so we should just let him maybe jump in here.
Well, Jared, I completely agree with you that this is one of the very tough problems. And, you know, you can in your imagination and imagine something that sort of handles each molecule as it's moved, but I can't think of a good way to do that. I think one of the tools that we might be able to make to separate the levels of metabolites from their fluxes. So when the level changes, if a level goes up, it could be because of increased synthesis or decreased degradation. And one possibility is that if we use things like Vomcy's enzyme, the LB-NOx, to if we could control that chemically to turn it on and off quickly, then we could make a step change in either synthesis or degradation, and that would help us disambiguate those two things. But in general, I think you're right. I think the mass spec tools are just so far superior to the C13 tracing that that's likely to be what we're limited to for flux measurements in general.
Great. I see Ralph raised his hand. Ralph, I'm sorry, I owe you a question from the previous talk. Go ahead now.
Oh, no, that's fine. Well, I just wanted to say, I mean, you know, I think the thing that's closest right now is the ability to actually image the transfer of carbon-13 from one molecule to the other. And so this is used clinically now in hyperpolarization techniques. That technology would have to massively improve to be useful in the type of scales that we're talking about here, where you need to have much better spatial resolution than what we currently get. And you wouldn't be able to flood the system with a massive amount of substrate, which is what's being used right now. But if those technical challenges could be improved, and this would be one way that you could actually detect like real flux or let's just say enzymatic activity in an intact system.
Fantastic point. I want to follow up on that probe approach. probes can be really subcellular and very sensitive, but it actually also has a property of it binds to its target, right? If you're measuring metabolite, it just binds to that metabolite. So potentially it can be a dual agent. It can be used to manipulate that metabolite level at the same time report that metabolite level. The challenge here is how do we separate the two, and can we use it for both, or we use it for one versus the other?
Yeah, I mean, I think we usually think about that in terms of unintentionally perturbing metabolite behavior by overexpressing a sensor. And I think that the comfort we can take is that the concentrations of the molecules to which the metabolites are ordinarily binding are usually quite high. So for NADH, NADH is tremendously highly buffered in cells by binding to all sorts of metabolic enzymes. And so, you know, I think we have a straightforward way to test just by varying the concentration of the sensors. We have a straightforward way to test whether we're perturbing the behavior. But I think in terms of manipulating deliberately, I think that would be a difficult way to do it. I mean, maybe we could, you know, in the spirit of Zolt's, you know, metabolite knockouts, we could overexpress a binding protein. But I think it would be cumbersome. And my guess is that the cell will adapt the free concentration to what it intends the free concentration to be.
Totally, that is a challenge.
Can I just say, I think that's one of the principles that I think, well, we've known forever, but I think at least I'm relearning, is that the metabolic network is not only complicated, it's also exceedingly dynamic. And the reason you can't knock out serine is not just because you can't knock out serine, because if you tried, the cell would figure out a way to make serine again. And then the rest of the network would be adapted to making serine in some new way. And so I think that's one of the other challenges we face, is that this sort of precision manipulation is very hard to do because things re-equilibrate. And you change one thing and something over there changes in response to that. And you don't know necessarily. I think there's a question from Mark.
Yes, Mark, go ahead.
You're still muted, Mark.
I'm standing far outside this field, so that may mean that I have great perspective that you don't have, or that I'm completely ignorant of everything. So somewhere in between. First thing I'm surprised at is no one has mentioned pharmacology, and as a perturbation method. And clearly that has a temporal element to it, with photoactivation or inactivation. It can even have a spatial component to it. So that's one issue that I've wondered a lot about. Also, non-specificity is a problem, but not every drug is non-specific in the same way. So, you know, by enough of this, you can piece together what the nature of the non-specificity is. And the second thing, which hasn't been mentioned at all, and again, it just shows how far away I am from everything here, is essentially no reference, or almost no reference, to a lot of work that's done in microorganisms, and where there's in fact less of the phenomena that you want to study, but there's a lot of principles that are really being established here. And so, so I just posed that question to somebody or all of you as to why I'm getting this impression and what's wrong with that impression, or are we really missing something here?
I'll tackle the pharmacology, Mark, which is that it's a huge tool, and it's something that we always use in combination with the biosensors to manipulate things. I think the limitation on looking at fluxes, using it to look at fluxes, is the speed, right? Because you wash in pharmacophores, they have to cross the cell membrane, and you know the phenomenon Jared mentioned about the system adapting is a big problem. So I think you know the quicker you can do it, we're trying to make optically engaged LB NOx enzyme, for instance, you know, something we can flip on and off with light as quickly as we can get the light there. I think the problem is that as clever as we want to be, metabolism is much more clever a lot of the time.
I would say that what Mark says is true, and that drugs are faster than genetics, right?
Oh yeah.
Even, you know, knockdown or acute knockout. And so I agree with what Gary said, that it is used extensively. None of us talked about it, but I think it is used extensively. And I also agree with Mark that model organisms are a great place to study. I mean, the cell biology of metabolism, I think, is to some extent recapitulated in simpler systems. And I think I'm a believer that it's very important to study it in those systems because we can do it at a resolution that you can't do in a whole organism. But obviously the integration with other cells is.
I mean, one way around the slow behavior of drugs in terms of diffusion and lots of other things is really photoactivation. And you know, I wonder how much that's been pursued, because you could diffuse in something which is inactive and photoactivate it. You could do that with drugs, but you could also do it with enzymes to some degree. And it would seem that the relaxation kinetics that occurs after that, spatially and temporally, might be very informative.
I just want to add to the pharmacology point. I think it is really powerful when we have good drugs that specifically target an enzyme that we're interested in. It's just not that common that we do have a very good drug for something we're interested in. And in Josh's lab, we have developed a few of these drugs, and they have been incredibly powerful in understanding metabolism of those pathways. So I do think if we develop more of those very specific drugs, it would be useful.
So one thing about that, we've done work with protein kinases and inhibitors and machine learning. And the combination of those three things allows one to essentially infer activities, even if they're non-specific, as long as you know from other experiments what the pattern of non-specificity is. And so, you know, I would hope that people might not be too afraid of that, even though I understand, because I think there may be ways around that.
Can I make one quick comment on the notion that I think everybody's brought up of how quickly the metabolic network resets itself to any sort of intervention, which I think is a really important concept. But it's actually, you know, it's both a problem and an opportunity. I mean, I think the same is true of genetic modification. You make a knockout of a particular gene in a mouse, you don't get always the phenotype you think you're going to get because there's a ton of feedback looping that gets activated. But then you study that feedback looping and you learn a lot about how and why that feedback looping is activated and the pathways that led to it from your original intervention. The same is true of modulating metabolic pathways and metabolites. You know, what happens afterwards is where you learn.
If you're lucky, you can see the effect of the acute intervention, and then you can see separately the adaptation of the system to it. If you're unlucky, you just see both smushed together.
Yes, yes. The kinetics of it are challenging, there's no doubt.
I think there's a lot of room for computational approach to address those adaptations, and especially at the system level.
Yeah.
There has been some work, Mark, on photoactivatable ligands, and even Bob Balaban has gotten some very interesting results at the NIH by literally photo bleaching NADH and then looking at the subsequent relaxation. So yeah, it's not, it's something that's being actively pursued.
Good.
So we've had a great discussion. I think we've a little bit over time, but not too much. And so I'll end this here and thank everybody for fantastic talks, a very inspiring discussion. And so I get this back to Ginny.
Wonderful. Thank you all. Let's take a 5-minute break and resume at 2:10 p.m. Eastern.